Adaptations for transport in animals. So first of all, wait, I think I've already gone through this one. Oh no, I'm getting confused with a different one. Right. I was confusing it with adaptations for gaseous exchange, because it is a very, very similar topic to that, because that would be 2.2 and this is 2.3. Yeah. So first of all, features of a transport system. You need to know, for animals, they need a medium to carry the materials around their body. Otherwise, they'd just be floating around. Wouldn't really be any way for them to transport the materials if there wasn't a material for them to transport them in. And they need a pump to be able to push that material around for the moving, well, what would be the blood a lot of the time. And they also have valves, so it flows in a specific direction. It prevents backflow, basically. Occasionally systems will have a respiratory pigment, like invertebrates and some invertebrates, they have that, but not in everything. The purpose of respiratory pigment is it increases the volume of oxygen that can be transported around the body, so it's just a bit more efficient. And they also need a system of vessels with a branching network to distribute the transport medium to all parts of the body. So, if you need something in the muscles, and you need vessels to be able to transport it to the muscles. So there are two different types of systems that you have. You can have open systems and closed systems for circulatory systems. Open circulatory systems, the transport medium doesn't move around the body in blood vessels. It just bathes in the tissues, which is a disgusting way to describe it, but yeah. It's usually held in a large space that's called, a large space in the body cavity called the hemocell. Insects have an open blood system. They don't really need vessels to pump their blood around, the vessels don't pump the blood, but they don't need vessels because they're very, very small. And they have this long dorsal shaped heart, it's like a tube, which runs all the way through the length of their body. It does pump blood, but it's at quite a low pressure into the hemocell, and then the materials are exchanged between the blood and the body cells, and the blood slowly returns back to the heart, and open circulation starts again. It's the oxygen in insects, in flies and stuff, it goes straight to the tissues from the tracheae, so it doesn't need... It doesn't transport oxygen, and it doesn't need a respiratory pigment. So here you can see the main vessel, which the blood is kept in, and it's just, yeah. So basically this diagram is showing you how it works. So this would be the heart that runs right down the length of the body, and it pumps the blood forwards, and then the blood is pumped into the main artery. There's only one, which is by here, and then it opens up into the body cavity, flows back through the body cavity, and then returns to the heart through the valves. Closed circulatory systems. You have a few more examples of these rather than just flies. You have, there's two types, the single circulation and double circulation. Single circulation, the blood only moves through the heart once in its passage through the body. The name is pretty self-explanatory. An example of that would be the earthworm. So the blood moves forward in the dorsal vessel. I don't know if you can see on this diagram, but that would be the dorsal vessel with the blood flowing forwards. And then you also have blood moving backwards in the ventral vessel, which is by here. Sort of not the best diagram, but you can see there's a little line there showing the ventral vessel. Yeah. You have these 5 pairs of pseudohearts around here, which are just thickened muscular blood vessels, and they pump blood from the dorsal to the ventral vessel, and just to keep it moving around. Another example of single circulation would be in fish. So the ventricle of the heart pumps deoxygenated blood from the gills, where its pressure falls. The oxygenated blood is then carried to the tissues, and from there the deoxygenated blood returns back to the atrium of the heart, and then the blood moves to the ventricle and the circulation starts again. So you can see the diagram here. It's not too complicated compared to the double circulation. So it's where blood passes through the heart twice in a circuit around the body. Mammals have a closed circulation system, and the blood is pumped by a muscular heart at a higher pressure. Usually, mammals, well, not in all cases, but, generally you can get larger mammals than other things. Like these examples, you have earthworm, fish, and then this would be a human body as an example. You need more pressure you have a lot of distance to push it through, so that's why, and it also needs to go through the heart twice as well. So lots of high pressure blood being pumped around the body. So it's a very rapid flow rate through the blood vessels. Organs aren't in direct contact with the blood, but they're bathed in tissue fluid, which seeps out of the capillaries. The blood pigment, hemoglobin, is the thing that carries the oxygen in the blood, and the blood pressure is reduced in the lungs, and its pressure would be the blood pressure is reduced in the lungs, and its pressure would be too low to make the circulation efficient in the rest of the body. So, because of this, because you have the lower pressure in the lungs, that's why it goes back to the heart, and then it pumps it through the heart again, so that gives it enough pressure for it to be able to be pumped to the rest of the body, so then materials can be delivered quickly to the body cells. Because if they didn't pump through back again, it would lose all its, if you have a diagram here. So, the blood would go around the body, and from here, I think this one's better. So from the pulmonary artery it goes to the lungs, but as it gets to the lungs, then the pressure is reduced, so it needs to go back into the heart and then get pumped out of the aorta, so it has enough pressure to be pushed around the rest of the body. Because, out of the pulmonary artery, it doesn't have the same amount of pressure as out of the aorta, because that's only going to the lungs, so it doesn't have enough pressure to begin with. So if on its journey it loses even more pressure, it wouldn't be able to move quick enough through the rest of the body. So that's why you have the double circulation system. So, in the double circulatory system, the pulmonary circulation serves the lungs. Pulmonary is always to do with lungs, so just knowing that is helpful sometimes, because you don't need to remember a word you don't understand. If you understand the word, it's easier for you to remember names of things. So the pulmonary circulation serves the lungs. The right side of the heart pumps deoxygenated blood to the lungs, and then oxygenated blood is it returns from the lungs to the left side of the heart. The systemic circulation serves the body tissues. So the left side of the heart pumps oxygenated blood to the tissues, deoxygenated blood from the body returns to the right side of the heart. And then in each circuit, the blood passes through the heart twice, once through the right side and once through the left. The double circulation of a mammal is more efficient than the single circulation of a fish, as oxygenated blood can be pumped around the body at a higher pressure. So it just means it's moving faster. The nutrients and gases are being transported to the muscles and wherever they need to be at, quicker, at a faster rate. So you need to know about 3 different types of blood vessels. So arteries, veins, and capillaries. There are more types of blood vessels. There are actually 5, but the 3 main ones for, basic knowledge is arteries, veins, and capillaries. Arteries and veins have the same basic 3-layered structure, but it's the proportions of these layers that are different. So the innermost layer, the diagram in the book wasn't very easy to see on camera. Everything's very blurry on this camera. So I did a badly drawn one. Circle's not very good, it's more of an oval, but you know, oh well. So what you need to know, innermost layer is the endothelium, which would be this bit here. And that is 1 cell thick, and it's surrounded by the tunica intima, which would be... I've drawn it quite big. I drew it wrong as well. So this would be the endothelium, as I said, and then there's a layer surrounding it there. That's called the tunica intima, which it's a smooth lining. I think both are. I think the endothelium is a smooth lining, which reduces friction with a minimizing the resistance to blood flow. So the blood flows faster with less friction through your blood vessels. The middle layer, called the tunica media, that would be this one here, contains elastic fibers and smooth muscle. So it's thicker in the arteries than it is in veins, because arteries, it's a higher pressure there, and the elastic fibers in it allow it to stretch to accommodate the changes in blood flow and pressure, because you know, in arteries it's just going away from the heart, so it's quite high blood pressure. Also, at a certain point, because there's elastic fibers, they also recoil, which pushes blood further through the artery, so it again speeds up the movement of blood, and that is why you can feel pulse. What it basically does is, not only does it just speed up how blood moves in the body, it also maintains the blood pressure, because well, you know, if it's moving further from the heart, then ordinarily, without this, the blood pressure would reduce, but because of the elastic recoil, it maintains the blood pressure as the blood is being transported away from the heart. And then you have the outer layer, the tunica externa, which is, you know, this bit. And that contains collagen fibers, which resist overstretching, because you don't want your blood vessels to be stretching too much. Now, the difference between arteries and veins, in the arteries, because they carry blood away from the heart, they need to be able to withstand the pressure derived from the heart, and that is why the tunica media and tunica externa is a lot bigger. Look in this diagram. It's a bit hard to see, but this is the artery and this is the vein. You can see the layers are a bit thicker here. And the vein's lumen, which is just the hole in the center. That's usually what they call any sort of the hole in the center of something in biology. The reason why they have a larger diameter lumen, I think it's just because the walls of the veins are smaller, so you have more room for the lumen. And they have thinner walls because they don't need so much muscle, because it's not so high in pressure there. So as I said, the blood pressure and the flow rate are lower. For veins above the heart, the blood returns to the heart by gravity. Veins also have these semilunar valves along their length, which ensures blood flow maintains its direction, so it prevents backflow. These aren't in arteries, other than at the base of the aorta and the pulmonary artery. They're not needed so much in the arteries because it's such a high pressure, it doesn't really risk too much backflow, but they still have them at the base of the aorta and the pulmonary artery. And if those ones don't work, then it can cause, it can contribute to varicose veins and heart failure. So valves are pretty important. So this is basically how they work as a diagram. So when they're the blood passes through the vein, and the valves open because of, the blood forces them open. But when there's more pressure downwards, then it shuts the valves, because, you have pressure here and here, which just forces them shut. Now, capillaries are virtually everywhere in your body. They're a very, very vast network, and they go to all tissues and organs in the body. The blood from capillaries collects into venules, which take blood into the veins. I'll just talk through the different types of blood vessels one moment. So, essentially, you have the arteries, and what they do is they branch into smaller vessels called arterioles. So that's my brief drawing of an arteriole. And then those further branch into capillaries. There would be a lot more capillaries than I've drawn. They would be also smaller than I've drawn, obviously. And then those go into venules, which then go to veins. And it's not the best diagram ever. Most of my diagrams are quite bad. But, that's just basically the order of things. It would be a bit more of a complicated thing in your body, but this is just a general overview of them. So, yeah. So the blood from capillaries collects into the venules, which take blood into the veins, which then return it to the heart. Loads of capillaries, they're numerous and highly branched, which means there's a large surface area for diffusion, which is a lot more efficient than, other methods. Capillaries have thin walls, which are only one layer of endothelium on a basement membrane. This also helps with diffusion. And pores between the cells makes the capillary walls permeable to water and solutes, though, like glucose. So the exchange of materials between the blood and the tissues is allowed to take place. Because capillaries have such a small diameter, when you go to them, when the blood flows to the capillaries, the blood flow slows down, because, there's not enough room for it to be moving so quickly with the same volume of blood. But because there are so many capillaries in the capillary bed which reduce the flow of blood, it means there's a higher contact time between, I wouldn't word it like that. There's just more time for diffusion to take place, so it means that more materials have a chance to move into the surrounding tissue fluid. There's a section on the heart, which, as you know, pumps to circulate blood around the circulatory system. I don't think you need to know much about this paragraph. You could think of the heart as two separate pumps, one dealing with oxygenated blood and one with deoxygenated blood. If it might make it a bit easier to think about. There's cardiac muscle in the blood, which is a specialized tissue with myogenic contraction, which means it can contract and relax rhythmically of its own accord, so you don't have control over it. And the heart rate is modified by nervous and hormonal stimulation. Unlike voluntary muscles, cardiac muscles don't tire, which is very useful, because if your cardiac muscles got tired, well, then your heart would want to stop, wouldn't it? And that wouldn't be really useful for you. So you have some diagrams here. This one is a lot easier to read than this one, I think. But yeah, so once I'll go with from the aorta. Blood moves out of the aorta to the body and organs, which then, goes around the body and organs, and then it moves back into the vena cava. And then it goes to the pulmonary artery, to the lungs, gets oxygenated, and then it goes back to the pulmonary vein. So pulmonary artery and pulmonary vein, because it's pulmonary and pulmonary, that just means relating to the lungs. So you know that these are both part of the oxygenation of the blood. it's going to or moving away from the lungs. And the way I remember arteries or veins, arteries away from the heart, so the pulmonary artery would be to do with the lungs, away from the heart, so surely towards the lungs. There are also valves in the heart. There's bicuspid and tricuspid valves. So there's a bicuspid valve there between the left atrium and the left ventricle. When you're looking at diagrams, the left is always on the right because it's like you're viewing another person's heart. So this would be the left side and this would be the right side. The left side is oxygenated, the right side is deoxygenated. So yeah, you have the bicuspid valve on the left and the tricuspid valve on the right, and they're between the atrium and the ventricle. And you also have, I believe they're called lunar valves. Sorry, semi-lunar valves, which are by here. So it's sort of between the ventricles and either the pulmonary artery or the aorta. What these do is they just prevent backflow back into the heart. Yeah, as any other valve would. So the cardiac cycle is the sequence of events of one heartbeat, which lasts about 0.8 seconds for the average person. And then there's, in the cardiac cycle, there's alternating contractions and relaxations. So contractions are systole, and relaxations are diastole or diastole, depending on how you want to say it. And there are 3 stages. So there's atrial systole, ventricular systole, and diastole. Atrial systole is where the atrium walls, these ones, contract, and the blood pressure in the atria increases, and that pushes the blood through the tricuspid and bicuspid valves down into the ventricles. So these contract and push blood down into these. And then ventricular systole is where the ventricle walls contract and increase the blood pressure in the ventricles, so these are now pushing. And that forces the blood up through the semilunar valves, which are open because if the blood's moving up in the direction it's meant to be, they are open, so they can move through the valves, and then out of the heart into the pulmonary arteries, and pulmonary artery, there's only one of them, and aorta. And the blood can't flow back because of the bicuspid and tricuspid valves being closed due to the rise in ventricular pressure. And yeah, the pulmonary artery carries deoxygenated blood to the lungs, and the aorta carries oxygenated blood to the rest of the body. Diastole is when the ventricles relax, and what that does is that increases the volume of the ventricles, so the pressure in the ventricles fall. This is what risks blood in the pulmonary artery and the aorta from flowing backwards into the ventricles, which is why you have the semilunar valves there. So the tendency of backwards flow causes the semilunar valves at their bases to shut, preventing re-entering of the ventricles. And the sound made when the beating heart sound is the sound of the atrioventricular valves and the semilunar valves closing. Yeah, and the atria also relax during diastole, so the blood from the vena cava and the pulmonary veins can enter the atria, and then it starts over again. So during this process, the atria contract at the same time, and the ventricles contract together at the same time as well. So the 2 sides of the heart work together. And yeah, you also need to know that atria walls don't have as much muscle because the blood only has to go to the ventricles. You can see that here in this diagram. There's not much muscle around the atria because it's only pushing it to this area. It doesn't really need the muscle in that area. Equally so, one side of the heart's muscle is thicker than the other sides because the left side has to pump blood all the way around the body and organs, whereas the right side only has to pump it to the lungs, which is it's a lower distance. Lower distance? Smaller distance. There is a part on the contraction of the heart, the cardiac muscles, to do with the electrical signals. So, contraction of the cardiac muscle is myogenic, meaning the electrical stimulation for the contraction originates from the muscle, and the wall of the right atrium has a cluster of specialized cardiac cells called the sinoatrial node, or SAN you can shorten it to, that acts as a pacemaker. So a wave of electrical stimulation arises at the SAN and spreads over both atria, and that makes them contract together. And because the ventricles they're insulated from the atria by a thin layer of connective tissue, except for another specialized cluster of cardiac cells, which are called the atrioventricular node, or AVN. The electrical stimulation only spreads to the ventricles from that point, and the atrioventricular node introduces a delay in the transmission of the electrical impulse, so that means that the ventricles contract after the atria do. And yeah, the slight delay of the wave of electrical activity in the AVN ensures that the atria are fully empty before the ventricles contract, which, is important, otherwise my blood would be going all over the place. And that atrioventricular node, or AVN, passes the excitation down the nerves to the bundle of His, which is right at the apex of the heart, which just means the bottom by here. And the excitation is then transmitted to the Purkinje fibers in the ventricle walls, which carry it upwards through the muscles of the ventricle walls. So the sort of movement that the ventricle walls make when they're contracting is like that. Very, very close. Like this. It sort of pushes it from the bottom to the top, so that everything is out. Because if you just sort of squish it like that, some might be moving down. It wouldn't be as effective in terms of everything, really. It would put extra pressure on the walls of the ventricles as well, so it's important that it pushes the blood up like that, because it empties the ventricles completely. So you need to be able to read electrocardiogram traces. So first of all, you have the P wave, which is the first small little part of the trace, and it shows the voltage change generated by the sinoatrial node, or SAN, which causes the contraction of the atria. Because they have less muscle in the atria than the ventricles, the P wave is quite small. My dog is barking! The time taken between the start of the P wave and the start of the QRS complex is called the PR interval, and is the time taken for the excitation to spread from the atria to the ventricles through the AVN, or atrioventricular node. And then the QRS complex, this bit, shows the depolarization and contraction of the ventricles. The ventricles have a lot more muscle than the atria, so as you can see, the amplitude is a lot bigger than that of the P wave. And then the T wave over here shows the repolarization of the ventricular muscles, and the ST segment lasts from the end of the S wave to the beginning of the T wave. So that would just be, ST segment. And then the line between the T wave and the P wave, which would be the second heartbeat, so between the first heartbeat and the second heartbeat, is the baseline of the trace, and it's called the isoelectric line. So there are some things that ECG waves can suggest if they're analysed. So you can read the heart rate, you can calculate the heart rate from the trace by reading on the horizontal axis, so this bit. So this would be about 0.6 seconds long. You just look from the beginning of the P wave to the the rhythm can also be shown by the regularity of the pattern of the trace. So if a person has atrial fibrillation, a person with atrial fibrillation has a rapid heart rate and may lack a P wave. A person who has had a heart attack may have a wide QRS complex, so this, rather than being like this, it would be a bit more like that. A person who has enlarged ventricle walls may have a QRS complex showing a greater voltage change and changes in the height of the ST segment, so this bit. And the T wave may be related to insufficient blood being delivered to the heart muscle, which can happen in patients with blocked coronary arteries and atherosclerosis. Yeah. So the length of the cycle is just the time between equivalent points on the trace. So if you look at the R to the R, so that would be from 0.3 to, it's in between 1.1 and 1.2, and then from that you can figure out the length of the cycle. You also need to look at pressure changes in the blood vessels, which can be shown on this graph. So the blood pressure is highest in the aorta and large arteries, and it rises and falls rhythmically with the ventricular contraction. And then the friction between blood and blood vessel walls and the large total surface area causes a progressive drop in the pressure of the arterioles, despite their narrow lumen, although their blood pressure also depends on whether they're dilated or constricted. And then the extent of capillary beds further reduces blood pressure, and fluid leaks from the capillaries to the tissues. So that would be around here. You can see that the graph's starting to slip down. In the arteries and capillaries, the higher blood pressure, the faster the blood flows through. The faster the blood flows. So both pressure and speed fall as the distance from the heart increases. Veins aren't subject to pressure changes derived from the contraction of the ventricles, so their blood pressure is low, which would be this part of the graph. Veins also have a larger diameter lumen, so blood flows faster than in capillaries despite the lower blood pressure. And then blood does not return to the heart rhythmically. Its return is enhanced by the massaging effects of the muscles around the veins. So this section of the graph would be where the blood's going to the heart and lungs. So this is left ventricle, arteries, arterioles, capillaries, venules, and then veins, and then the heart and the lungs. You need to know what makes up blood. So blood is a tissue made up of about 45% cells and 55% plasma. Red blood cells, which are also called erythrocytes, are red because they contain a pigment hemoglobin, the main function of which is to transport oxygen from the lungs to the respiring tissues. And red blood cells are strange cells in two ways. They don't have a nucleus, which maximizes the space for hemoglobin, which means more oxygen can be carried, and they also have this biconcave disc shape, so it sort of dips in both sides of it, and this is what it looks like at a different angle, which increases the surface area rather than just a plain disc, so it means more oxygen can diffuse across the membrane, and the thin center makes them look paler in the middle in some pictures. If you look on this graph, it's a bit hard to see. In fact, it's showing it to you the other way around. So, on a microscope, red blood cells might look a little bit like that, but that's just because they have a thin center. So, yeah. And what it does is just reduces the diffusion distance, which means gas exchange is a bit faster than it would be otherwise. You also have white blood cells, which can be, which are called leukocytes, and they're larger than erythrocytes. And there are two main types of them, so granulocytes, which have a granular cytoplasm, and lobed nuclei, they're phagocytic. There are also granulocytes, or lymphocytes, which is, I just say lymphocytes, and they have a clear cytoplasm and a spherical nucleus, and they produce antibodies and antitoxins. So basically, the prefix phago means to eat or devour, and cyte just means cell. So phagocytic just means it's a cell that eats or devours other cells. So what they essentially do is they just sort of... Oh my gosh, my dog! So what they do, granulocytes, they basically just... they just sort of surround bacteria, and they... well, just... they don't... not dissolve, well, they just eat them. It's just the simplest way to put it. They just eat them, okay? They break them down. They digest them. And as it says here, lymphocytes just produce antibodies and antitoxins. Plasma is mostly water, and it contains solutes such as food molecules like glucose, amino acids, vitamins, and mineral ions. And they also have waste products like urea, hormones, and plasma proteins. And also blood clotting proteins and antibodies. It just has a lot of stuff in it. It's pale yellow, and it also distributes heat as well as transporting all of those materials. Now, with the transport of oxygen, you need to use very specific language. So, instead of saying that hemoglobin picks up oxygen or releases oxygen, you need to say disassociates disassociates and associates, or loads and unloads. So, hemoglobin binds to oxygen in the lungs and then disassociates or unloads it in respiring tissues. You have a little equation there, O2, HB, HB for O2, because hemoglobin can carry 4 oxygen. To transport oxygen efficiently, hemoglobin needs to associate readily with oxygen where gas exchange takes place, so at the alveoli, and also readily disassociate from oxygen at the respiring tissues like muscle. So hemoglobin can perform seemingly My dog is barking again. Basically, what hemoglobin can do is change its affinity for oxygen by changing its shape, which, makes things easier. Please be quiet, dog. Oh, I'm dropping things. So each hemoglobin contains 4 heme groups, and each heme group contains an ion of iron, so Fe2+, 1 oxygen molecule combined to each ion. Really horrible phrase, ion. It sounds like I'm saying the same thing. So 4 oxygen molecules combined to each hemoglobin molecule. So when the first oxygen molecule attaches to the shape of the hemoglobin molecule, I'll do a little diagram. Usually they're sort of drawn as a cross. So 1 Fe2+ joins to the hemoglobin, draw it like that, and that changes the shape and makes it easier for the second ion to bind. Sorry, that should be an oxygen molecule. What am I doing? Oxygen! The oxygen... I'm just doing a different diagram. Have your hemoglobin. I'm drawing it like this because it changes shape, but it's easier to just draw it like this initially. So the first oxygen binds to the hemoglobin, and that makes it easier for the second molecule to attach because it changes its shape. That would be called cooperative binding because it allows hemoglobin to pick up oxygen very rapidly in the lungs. And, it's just cooperative. It's all working together. The oxygen molecule does not induce a shape change. What it should say there, the third oxygen molecule doesn't induce a shape change. The shape has already been changed, so it takes a very large increase for the fourth oxygen well, it takes a large increase in the oxygen partial pressure to bind the fourth oxygen molecule. Basically, the partial pressure of a gas is the pressure it would exert if it were the only one present. So, normal atmospheric pressure is about 100 kPa, and oxygen comprises about 21% of the atmosphere, so its partial pressure is 21 kPa. So, when a pigment is exposed to increasing partial pressure of oxygen, if it absorbed oxygen evenly, the graph plotted would be linear. But, so as in, it would be like oxygen in the atmosphere to binding, let's say, the rate of binding. I've written that really horribly. If each oxygen molecule if each oxygen molecule bound to the hemoglobin equally easily, then the graph would just be straight linear. But because of the way oxygen binding works, it doesn't work like that. Cooperative binding means that hemoglobin exposed to increasing partial pressure of oxygen is actually a sigmoid shape, which is an S-shaped curve. So at very low oxygen partial pressure, it's difficult for the hemoglobin to load oxygen, but the steep part of the graph shows oxygen binding increasingly easily. And then at high partial pressures of oxygen, the percentage of saturation of oxygen is very high. So you have a little curve there. This is what you'd expect it to look like, but it actually looks like this because of the way that cooperative binding works. So the oxygen affinity of hemoglobin is high at high partial pressure of oxygen, and oxyhemoglobin does not release its oxygen. Oxygen affinity reduces as the partial pressure of oxygen decreases, and oxygen is readily released, meeting respiratory demands. Although you wouldn't want to say released, what you want to say is it's disassociated or unloaded. Yeah, and the graph shows that a very small decrease in oxygen partial pressure leads to a lot of oxygen disassociating from hemoglobin. So red blood cells can load oxygen in the lungs where the oxygen partial pressure is high, so there's lots of oxygen there, and the hemoglobin becomes saturated with oxygen. The cells carry the oxygen as oxyhemoglobin to the respiring tissues, such as muscle, and there the partial pressure of oxygen is low because oxygen is being used up in respiration, so the oxyhemoglobin then unloads its oxygen, or disassociates it. Actually, both would work. It unloads/disassociates its oxygen because of the lower partial pressure in the muscle. Now, certain animals, or I don't know, the partial pressure of oxygen can affect how things, your affinity, your hemoglobin's affinity for oxygen. So in a fetus, the hemoglobin in the blood of the fetus needs oxygen so it can grow. So the fetus has hemoglobin that differs in the 2 of the 4 polypeptide chains from the hemoglobin of an adult, which means that fetal hemoglobin has a higher affinity for oxygen than the mother's hemoglobin at the same partial pressure of oxygen. So, if even when both the hemoglobin of an adult and the hemoglobin of a fetus are in the same environment in terms of how much oxygen's there, the fetus will still have a higher affinity for oxygen there because it needs it to be able to grow. Myoglobin is a muscle protein, and its oxygen disassociation curve is far to the left of hemoglobin. So you have a graph here. You can see partial pressure of oxygen and percentage saturation of hemoglobin with oxygen. So this would be adult hemoglobin. So the saturation it increases at a high partial pressure, and as you move along, that would be the fetal hemoglobin. So it has a high saturation of oxygen at a low partial pressure, which means it has a higher affinity. And then that would be that of a llama, we haven't covered yet. And that would be myoglobin. So it's really far to the left of the curve, so it's very high affinity for oxygen. So that means oxyhemoglobin only unloads its oxygen when the oxygen partial pressure is very, very low, like when you're exercising heavily. So myoglobin is described as an oxygen store. Lugworms live head down in sand or the seashore, so it's a very low oxygen environment. It has a low metabolic rate because it doesn't need, it doesn't move so much, and its hemoglobin has a dissociation curve to the left of human hemoglobin, which means its hemoglobin loads very readily, but only unloads when the partial pressure of oxygen is very low, which is the situation of its habitat. So it's just sort of adjusted things for the habitat it lives in. And then llamas live, at altitude. So oxygen partial pressure in the atmosphere decreases, which is quite significant for mountain animals, such as a llama. So its hemoglobin has developed a dissociation curve to the left of human hemoglobin, which means it's higher affinity for oxygen. So its hemoglobin has a high affinity for oxygen at all oxygen partial pressures, so it loads oxygen more readily in the lungs and releases oxygen when the oxygen partial pressure is low in its respiring tissues. So the effects of carbon dioxide concentration. If carbon dioxide concentration increases, hemoglobin releases oxygen more readily, because that would mean there's less oxygen in the environment, more carbon dioxide. And at any oxygen partial pressure, the hemoglobin is less saturated with oxygen, so the data points on the dissociative curve are all lower. This is described by saying the curve moves to the right. The shift in the graph's position is called the Boer effect. So carbon dioxide concentration affecting hemoglobin is the Boer effect, and it accounts for unloading of oxygen from oxyhemoglobin in respiring tissues, where the partial pressure of carbon dioxide is high and oxygen is needed. So at a given partial pressure of oxygen, when the partial pressure of carbon dioxide is higher, hemoglobin has a lower affinity for oxygen. Can't speak. So, transport of carbon dioxide is quite interesting. It's transported in three ways: in solution in plasma as hydrogen carbonate ion, and bound to hemoglobin as carbaminohemoglobin. Some carbon dioxide is transported in red blood cells, but most is converted in the red blood cells to hydrogen carbonate, which then diffuses into the plasma. So this is to describe the reactions in the blood cell. First, carbon dioxide. I'll draw a little diagram as I'm explaining this. Well, I've already done I thought it would just be easy to do a diagram first and then I could talk, rather than drawing it at the same time. So, carbon dioxide in the blood diffuses into the red blood cell, which is... I'm moving this back a bit. Ooh. So, this is the red blood cell, and this carbon dioxide diffuses into the red blood cell, and then carbonic anhydrase catalyzes the combination of carbon dioxide with water. So, carbon dioxide with water reacts with carbonic anhydrase, catalyzing that reaction, and that makes carbonic acid, which is H2CO3. And then carbonic acid then dissociates into a H plus ion and HCO3 minus ions. That should have a plus there. And then the HCO3 minus ions diffuse out of the red blood cell into the plasma. And to balance the outflow of negative ions in the red blood cell and maintain electrochemical neutrality, chloride ions diffuse into the red blood cell from the plasma, and that movement is called the chloride shift. And then the H plus ions, the positive ions, cause oxyhemoglobin to dissociate into oxygen and hemoglobin. So, I think, yeah, oxyhemoglobin there. And then the H plus ions combine with the hemoglobin to make hemoglobinic acid, which is HHB, and this removes hydrogen ions so the pH of the red blood cell does not fall. And then the oxygen diffuses out of the red blood cell and into the tissues from where oxyhemoglobin was there. And yeah, you need to know about some tissue fluids, intercellular or tissue fluid. So exchange between the blood and body cells happens at the capillaries. Plasma solutes and oxygen move from the blood to the cells, and waste products such as carbon dioxide and, in the liver, urea move from the cells to the blood. Capillaries are well adapted to allow this exchange of materials. They have thin permeable walls, large surface area for exchange of materials, and the blood flows very slowly through the capillaries, allowing time for exchange of materials. Fluid from the plasma is forced through the capillary walls as tissue fluid, and then it bathes the cells, supplying them with solutes such as glucose, amino acids, fatty acids, salts, hormones, and oxygen. And then the tissue fluid removes waste made by the cells. The diffusion of solutes in and out of the capillaries relates to the blood's hydrostatic pressure and solute potential. So here is where the pink arrows are where useful materials like glucose and oxygen diffuse from the blood into the tissue fluid, and then the waste materials like CO2 and urea diffuse out of the cells from the tissue fluid, and then the tissue fluid diffuses into the blood capillary or lymph capillary. And then, you know, you can... and then it's taken elsewhere to be sorted out. You can see the forces acting on the capillary bed in this diagram. So at the arterial end, there's high hydrostatic pressure of blood, and the solute potential pulls water inwards, less than the hydrostatic pressure. And at the venous end, venous, I think that should have an I. The hydrostatic pressure is much lower as the fluid has been forced out of the capillary, and solute potential is a little more negative than at the arterial end. That's it really. There's definitions that you need to know, but I think I've already gone through those through the book. So yeah, that's Adaptations for Transport in Animals. There is another thing I didn't look at, but I would consider looking over the changes of pressure in the heart, because there's a lot of exam questions on them, so you need to be prepared to describe the pressure changes involved in the flow of blood. So the way it's been written down in my book is the pressure rises due to the atrial systole contraction. So the left atrial pressure, first of all, the pressure rises due to the contraction of the atria. So yeah, left atrial pressure, which would be the blue one. So it doesn't really change that much, but you know, it rises. And then it decreases as the blood flows into the ventricles. And then atrioventricular valve closes due to the ventricular pressure rising above the atrial pressure. Pressure then steadily rises as the blood is returned to the atria while the atrioventricular valve is closed. And then the atrioventricular valve then opens, and the atrial pressure drops as blood flows from the atrium to the ventricle. So that's just explanation of this line. For the left ventricle, the pressure rises slowly as blood is forced from the aorta into the ventricle. The ventricle contracts, and that causes the atrioventricular valves to close and the pressure to rise sharply until it's great enough to cause the semilunar valve to open, and the blood is forced into the aorta. So that's left atrium. And then the lack of blood in the ventricle causes the pressure to drop. When it drops below aortic pressure, the semilunar valve closes due to the backflow pressure in the aorta. So you can see it drops here below the atrial pressure. And then both valves are now closed, and the ventricular pressure continues to drop during diastole. When the pressure drops below the left atrial pressure, the atrioventricular valve opens, and the blood flow in the ventricles blood flows into the ventricle, causing a gradual pressure rise, which is by here. And then you also have the aorta. So aortic pressure, the pressure gradually falls following the previous heartbeat, by here. The semilunar or aortic valve opens, and the blood from the contracting left ventricle is forced into the aorta, causing a rise in pressure, which is here. The left ventricle relaxes, and the aortic pressure starts to fall, by here. The backflow pressure in the aorta causes the semilunar valve to shut, and when the aortic semilunar valve closes, the arterial walls recoil, which is from, elastic recoil, causing a brief pressure rise in the aorta. And they like giving you questions on those, so I would recommend looking over it, because it's a bit difficult to grasp, well, at least for me. So, yeah, try to memorize that.